Going to be a great follow-on to Ed's great talk about how pain is unreliable and we really don't understand it. We, by contrast, are at the very beginning of that 5-year adventure and trying to figure out what we can have as better biomarkers so that we can start to understand pain and how to improve our understanding and treatment of pain in fractures. So, this is an obvious statement, fractures hurt, we all know that, but it's actually kind of wanted to draw attention for a moment to some of the data on why it hurts in the different fibers and kind of just do a quick anatomy review. And so one of the parts is that there's both sensory and autonomic nerves in the bone. The majority of the sensory neurons are in the periosteum of the bone, and you have both A fibers and A delta and C fibers. And so that is the difference between the quick pain, which is the A delta fibers, versus that sustained chronic pain on the myelinated C fibers. And so just wanted to kind of put this diagram up there as a reminder of where the nerves are. And this is some really beautiful histology where you can actually see the nerves in the periosteum, but you see that the nerves actually penetrate all the way into the metaphysis of the bone, and so your bone is really innervated, but the ones that are really painful are in the periosteum. And so you all know this, opioids are the standard of care to treat fractures, both acutely to start, and part of that is from maybe poor evidence, but a lot of evidence or a lot of papers that have come out saying NSAIDs cause a delay in fracture healing. And I don't know if anybody's read this book, it's really interesting, but there's also been this propagation of like, there's just a little data on NSAIDs being bad, and so then it was, a big push to use opioids instead. And I just wanted to put up some facts that I find really interesting, which is 56% of patients, and this was in 2017, this paper just came out in 2020 in JAMA, but in 2017, 56% of Medicaid patients got opioids, 44 are privately insured. Some of these numbers are crazy. Femur fractures getting 47 pills of oxycodone with 3 refills. The kids one really bugged me. The average of, giving kids opioids right now, I don't know if you've heard, like Sesame Street even has some stories on how to deal with addiction for kids. So it's a big problem. But there aren't great solutions right now. We don't have good alternatives, and part of it is because we don't have good methods to understand and test pain. This was another part that was interesting. So if you're in the pain field, if you study pain, the definition of pain is chronic pain is 3 months or more of pain. And so actually a ton of fracture patients go on not only to have acute pain, but this chronification of pain. These are multiple different studies. Obviously people have different rates, but there's a pretty significant transition to chronic pain. So there's a few problems we have to understand. One, how to treat acute pain. Also, can we understand who and how and why you get chronification of pain in some of these instances? So one of the other problems is innervation is actually really important to the fracture healing process. And so if you don't have nerves, you actually don't get good fracture healing. And what's interesting is we all know, we've talked a lot about vascularization and the importance of vascularization in promoting healing, but the nerve comes in first. And that is something I think that is underappreciated. And then there's been a number of papers that talk about if you get rid of the nerve or block the nerve, you don't get fracture healing. You don't get good bone healing. Actually, you can also just get bad as part of the contributing factor to even osteoporosis. There was an anti-NGF that they tried for pain in osteoarthritis, and it actually caused worsening of the subchondral bone, and so acceleration of OA. So we know we can't just completely block neuronal signaling. And then some work out of our lab actually found that by contrast, if you give nerve growth factor, accelerate the nerves coming in, it can accelerate fracture healing. And what was really interesting is if you give nerve growth factor really early in the fracture, it doesn't work. But if you give nerve growth factor when you have a cartilage or a soft callus, that actually accelerates the conversion of cartilage to bone. And here's some histology and data to just show that we can quantify this in a mouse model, that giving NGF can actually accelerate bone healing. And it does this by accelerating the process of endochondral ossification, so accelerating cartilage to bone conversion, and reducing apoptosis. So the nerves help keep the stem cell niche and cells in the fracture alive. More data, this has been published if you're interested in the details. So we've kind of moved into this new area in the lab where we're really trying to understand pain signaling so that we can start to work on new therapies. And so everything's so confounding, but what we really got to is that no one has good data on what causes fracture-related pain, how it interacts with inflammation, and how you can eventually get a biomarker so we can end up with a tool like EDS that can maybe tell us how pain is working. So one of the reasons is we don't have a good way to measure pain in humans. We also don't have a good way to measure pain in mice, which is where we're going to be able to vet out new technologies and new therapies. So the ones that existed are these von Frey filaments, where basically poke a mouse's foot. They do work in some tests, especially like diabetic neuropathies, that sort of thing, but they don't work with a fracture because it requires the mouse to move its paw away. You can put a mouse on a treadmill. You can put a mouse on what's called a catwalk, which is a really thin bar that they have to walk up across. But this is just not going to work well in the early stages of fracture where a mouse can't walk at a gait or walk on a bar. So we were able to use an NIH grant to actually buy this new system that came out of Harvard called the black box. And the reason this has been advantageous is it puts a mouse in its natural environment, which is dark. They're nocturnal. They can smell people. So you put them in this box, and they can wander around, and you can get recordings of the mouse where you're not interacting with them, and they are not, you're viewing everything from the bottom, because from the top, as mice being prey animals, they will hide everything. So this is just a naive mouse, no pain, walking around, and you can see you get two things. You get its body position, you get rate, but you also get pressure. So this is just an example of like some of the outputs you get. And so we went out working with this pain specialist here at UCSF, Jarrett Weinrich, and we decided to just see if we could use the system to adequately distinguish differences in pain. So we took a mouse, we made a stable fracture with basically a little intermedullary pin, and then we also took that fracture and left it unstabilized. And we looked at male and female mice and wanted to see if we could tell the difference. So in the video on the top that's playing, you can see at 5 days after fracture, you can clearly tell which foot is fractured. The toes are curled, they're not putting as much weight on it. By day 11 or 10, I think it's cut off on my screen, I can't exactly see, but it's starting to put some weight back on it. And by day 19, you're really starting to see the mouse move around. So we wanted to basically quantify all of this, and we had this poor student, graduate student, her name's Molly, she's really great, and she basically watched mouse TV for a long time to figure out what behaviors were different, and we basically used AI, a method called Deep Lab Cut, to train the system to understand what features of the mouse we could quantify in a much more robust way. So this is just an example of some of the data that came out of it, and you can basically see this is in female mice, the naive, the stabilized and unstabilized, and you can see that kind of as expected, the stabilized fractures start to put more weight earlier than the unstabilized fracture. And this is a heat map just to show you that we collected lots of different data and kind of came up with a series of things that we thought were important. And again, the conclusion here was the level of pain, or the level of functional deficit in an unstabilized mouse was much bigger than the stabilized mouse. They recovered more quickly when the fracture was stabilized, and it wasn't as much pain or as much functional deficit. And then we use this graph theory, which is this concept to be able to take all the data that we collected across all the different metrics, and we could see that males and females clustered into different areas, and also the fracture recovery clustered into different areas. And the reason we did this is we had all of those different metrics that we were looking at, but sometimes some things went up and some things went down, and we couldn't try to figure out like what was the most meaningful. So we used this technique of graph theory to try to come up with a unified metric of recovery. And so this is just showing you that we put all of that data and came up with sort of one functional recovery system that we think will help us screen for novel therapeutics or novel opioids. And you can see here that, again, stable fractures are not as severe and they heal a little bit more quickly. And you can see there are some different patterns between males and females, not as significant as we thought they could be. So the next question was like, okay, so we now have this functional readout, but what's happening in the transmission of pain? And so this project is being taken on by a postdoc in the lab, Masataka Toi, who's here today, and he's doing this really technically challenging system to try to understand how the pain is going from the fracture to the dorsal root ganglia. The dorsal root ganglia is right off the spinal cord. It's collecting some of the pain signaling and triggering that to the brain. So he made the stable fracture, and then he injected a retrograde tracer that's fluorescent red, and so that we could actually look at when you have a tibia fracture, where is it going in the spine? And he showed that this is the top row is a sham-operated animal, and the bottom one is a fractured mouse with the pin in it. And basically, he shows that it's L3, 4, and 5 that are really receiving the signal from a tibia fracture, and that with tibia fracture, the L4 and L5 become more activated. So it's actually very interesting that this data is not really something that you can find very easily right now. And so this is some of the first data using a tibia fracture, a full fracture, that looks at this. And what's also interesting is using this stain, the green stain is for the marker ATF, and it's a marker of nerve injury. And what he can show is that he can see nerve injury all the way out to 21 days in the dorsal root ganglia, even though functionally the mouse is starting to recover by day 15 in 2 weeks out. So we can actually see pain and neuronal damage in the DRG for much longer. So where we're going with this is actually, this is another really cool experiment where we basically put a camera into the mouse's brain, and they can live with this camera in their brain for like they can actually live for a year with it. And this is allowing us to look at the neuronal signaling up to the brain, and we can look in the pain center and understand how that is going from the fracture to the dorsal root ganglia back into the brain, so that we can try to see what's causing pain. And basically in these little neurons in the video on the right, every once in a while you see some flashes of light, and that's calcium signaling causing the pain in the brain. So that's where we're going, and I'll just end with a couple reminders. Obviously, bone breaks hurt, but the nerve's really important, so we can't just get rid of the nerves. It's important to healing. And so we're trying to get better systems so that we can phenotype pain in the lab, with the goal of understanding what causes chronic pain, testing novel therapeutics, and then also testing non-opioid alternatives. And I think many of you are coming over for the course this afternoon, or were there earlier. So my lab is on the second floor of Pride Hall. So if anybody is interested in coming up and seeing our basic science research lab, which is just one floor above where the course is at the new Pride Hall, I'll be happy to show people around. Thank you very much.